Okay, good morning, everybody, on this wonderful Saturday morning. Thanks for joining me. This morning, I'm going to be talking about the plant use at the Sheffield site. I am sure you have last couple years have probably heard Dr. Ed Fleming talk about the Sheffield site and the excavations that we've done on the site. And this year, I'm going to talk about the plant studies that I have done from the 2013, 2017, and 2015, excavations that we have done from the site. And so the Sheffield site is a multi-component site for a mound and habitation complex located on a sandy outwash terrace overlooking the St. Croix River about 10 miles north of Stillwater. The habitation part of the site is located immediately adjacent to and amongst a mound group of 26 mounds originally mapped by T.H. Lewis in 1885. Excavations by Lloyd Wilford and Peter Jensen in the village areas of the site identified significant amounts of Oneota materials and intact habitation features along with lesser amounts of earlier, middle, and late woodland materials. In 2012, Ed Fleming, curator of archaeology, received a legacy grant to be able to return to the site to process the old collection from 1959 and 1960 from Peter Jensen's work and conduct another excavation in the summer of 2013. This season, 10 cultural features were recovered and all soil matrix was collected for flotation and future plant inventory. After processing the 2013 collection, Dr. Fleming applied for a small legacy grant and for me to be able to process 195 soil samples and to analyze the plant remains from 7 of the cultural features that were recovered. In 2015 and 2017, Dr. Fleming returned to the Sheffield site with 2 field schools, and in those seasons, 17 more cultural features were recovered. In 2017, Dr. Fleming again applied for a large legacy grant to study the subsistence practices from the Sheffield site and Wilford site. In this grant, we were able to analyze 7 cultural features from the Sheffield site and 3 features from the Wilford site that were analyzed. So in the next couple slides, I'm going to go over the features that we analyzed during the subsistence project and a couple of the features that we did from the 2013 project. In block 1 that was first opened in 2013 was a 2 by 2 meter unit and was broken down into 1 by 1 meter units. Features 2, 3, 4, 6, and 10 and half of feature 5 were found during this season. Features 2, 3, and 5 were identified as fire hearths. Feature 5 was a 1 meter in length and 50 centimeters in width found at that time. Feature 4 is a mussel shell midden. Feature 6 is an amorphous area, and feature 10 is an ash and charcoal deposit. All of these features were used in the 2013 plant inventory and C14 dating for the site. In 2015, block 1 was expanded to recover the rest of feature 5, and feature 12 was also recovered and collected, but was not used for the plant analysis. Upon fully discovery, feature 5 was actually a 1 meter in width and length after full exposure. Block 5 was opened in 2015, and it was a 1 by 5 unit and broken into 1 by 1 unit, 1 by 1 meter units inside. Features 13, 19, and most of feature 18 were collected during the season. Feature 18 and 19 were used for the subsistence project, while feature 13 was not used in the current analysis. Feature 18 was identified as a storage pit and hearth, and feature 19 was identified as a storage pit. In 2017, a 1 by 2 meter unit was added to block 5 to collect the remaining culture deposit of feature 18. During the excavation, feature 21 was recovered. It was identified as a storage pit and was used for the subsistence project. Block 7 was first opened in 2015 as a 2 by 2 meter unit broken down into 1 by 1 meter units. Features 20, 22, 24, and 25, and 26 were collected during the 2015 season. Features 20, 22, and 24 were used for the subsistence project. Features 25 and 26 were not used for the analysis. Features 20 and 22 were identified as a fire hearth, and feature 24 was identified as an FCR concentration hearth with bone and charcoal overlaying a pit. In 2018, we returned and expanded the 2x2 into a 3x3 unit to gather the rest of feature 25. During the excavation, we uncovered an old excavation unit from Peter Jensen in the his 1960s dig. So I'm going to go into the methodology that we use for the plant analysis. So 100% of all the 360 soil samples that we collected from the 2015 and 2017 seasons that were collected were processed by my friends and coworkers, Mara and Taylor Brame, at the St. Croix Watershed Research Station with our new float machine during the summer of 2018. All the soil samples were floated and they were brought back to the Science Museum for further processing. Non-carbonized material, recent plant remains, plant roots, bark, and unburnt seams were removed and discarded. Material was sorted further down with the material was sorted further from 10 mesh, separating the wood and non-wood. Wood material was not identified below the 10 mesh. Non-wood material then was sorted into 18, 35, and 60 mesh screen size. Non-wood material includes seeds and other non-wood plant parts. Wood remains are those that come from branches and tree trunks of deciduous and coniferous trees. Dr. Ronald Shermer from MSU Mankato, a paleoethnobotanical expert, was consulted to analysis and identifying any problematic specimens that was needed. The wood or charcoal assemblage of the Sheffield site includes wood used for firewood in the village. The range of species present at the Sheffield site include tree species typical of the local environment surrounding the site today, and is what would be expected from the area during the time the site was occupied. Today, the floodplain forest and mixed hardwood forest dominates the landscape around the site. Likewise, vegetation estimated for the pre-settlement times include hardwood forests, oak openings, river-bottom forests, and prairie in the area, as indicated in Marshner's 1975. White oak, as an easily accessible and excellent fuel source, is the most abundant tree species at the Sheffield site. Following the oak abundance is white ash, white pine, maple, unidentified conifer, American elm, ironwood, box elder maple, red oak, dogwood, birch, hard maple, poplar and willow, elm, red maple, black cherry, hickory, and other are present, but they're in less quantities. I also like to apologize for my charts. They can be a little bit, there's only 2 of them, but I know they're a little bit big. So these are Ubiquity charts. Ubiquity are used to calculate for both in the wood and the non-wood. Ubiquity is a measure for the relative abundance of the plant taxa within the assemblage based on the presence and the absence. It is an efficient way to visually determine whether there's a particular species is widely or narrowly distributed across the sample or whether a co-occurrence exists. It is also an effective way to determine diversity among plant species within a single feature. Species with higher ubiquity numbers are more widespread than those with lower ubiquities. The result of the ubiquity calculated for the wood species are shown. Similar to the overall abundance, white oak of 1 was widely distributed, found in all features. Feature 5 and feature 24 have the highest number of diverse species present, which is not expected as both of these were hearth-like features. The rest of the features had very low on the presence of diverse tree species, which could be the account for the feature function. Of note, feature 20 contained a burnt terminal bud. The terminal buds are found on the end of branches that are typical of younger trees. The tree species that are the species that could not be identified. The buds are given a seasonal window as to when the feature was created and also a seasonal indicator to use of the Sheffield site. The time frame would be from mid-springtime, since this is when the tree begins to leaf out. We are going to move into the non-wood, and we're going to start with cultigens. Cultigens are a domesticated plant that have been selected to keep growing from year to year. It's spread from wild varieties. These plants are important staple for diets and culture of the people who take the time to nurture and grow them to use to feed their communities. The non-wood assemblage is composed of these remains that are from the non-woody reproductive parts of the plant. In some instances, though, a cultural heritage is speculative. Cultivated plants that are domesticated and cultivated in the past are those typical of Oneota habitation sites from the late pre-contact period. Common plants of Oneota are known to have cultivated include maize, squash, gourd, beans, plus oily seeds providers such as sunflowers and starchy seeds provides of goosewood, marsh elder, and little barley. The Sheffield site has all these present except for a little barley at this time. The Ubiquity Chart was used to show the absence and presence again for this for the non-wood plants for this site. Maize has the highest ubiquity of all the non-wood plants present at this site. It is present in 11 of the 13 features collected. Feature 22 from the 1960 sample was not included in this part, as it was just a charcoal sample from that from the 1960 excavation. The second highest ubiquity would be pop starch. If anybody is unfamiliar with what pop starch is present in 7 of the 13 features. Pop starch is created when starchy foods are cooked, such as tubers, maize, or rice, are cooked at high temperatures. Gelatinization occurs and can be preserved in the archaeological record. During heating, the molecular structures of the plant changes due to the interplay between heat, water content, and the starches, proteins, and lipids in the plant. Because of this, it is difficult to identify specific plants in the forms of pop starch. Sunflower seeds is third, squash ferns are fourth, and pigweed are second. They're next into the descending order of the ubiquity table. Moving down the table, we find more non-cultigens mixed in with some cultigen material. Non-cultigens are plant remains that have no evidence of domestication. This does not mean that these plants do not have in some way of an importance. Example, there are low presence of fruits or nuts at the Sheffield site. Some material could determine if there's a seed rain, if there's a low presence, such as like poison ivy that we have at the Sheffield site, or ragweed. That could be from seed rain, but it's undetermined since we have a low sample. Feature 5 had the most diversity plant species out of all the other features that were collected, with had 22 diverse plant species. Feature 2 was second with only 13, and feature 24 had 11. So my discussion and conclusion for the Sheffield site: we analyzed 8 fire hearths, 2 storage pits, 1 shell meadow, and 1 amorphous area that was analyzed from the site from over 3 seasons. The non-wood plant species from the Sheffield site combined do put into the line that was expected from the Oneota village from the 13 to 14 CE period. Comparing the Sheffield site to other areas such as the Red Wing region and La Crosse locality is important because even though our sample size may be small, it can help us with broad generalizations to help comparing and contrasting. The Sheffield site compared to La Crosse locality can be misleading since the La Crosse area has a long-standing and well-documented paleoethnobotanical research. Even though there are only 14 features at the Sheffield site to compare with hundreds that may have been analyzed in the La Crosse area, some generalizations can be noted. The variety of nuts and fruit that are present at the botanical assemblage in La Crosse compared to Sheffield is notable. This also can be the same for the Red Wing region. Both Sheffield and Red Wing both do not have this. They lack the variety and the quantity that La Crosse does have with fruit and nuts. Sheffield only has 2 grape seeds and 1 hickory nut fragment, and 39 acorn fragments. The McClellan site and Burnside School site in the Red Wing region have similar low frequencies and nut and fruits, such as the Sheffield site does. So far, tobacco is not present at the Sheffield site, and this is also similar with the single component sites in Oneota villages in the Red Wing region, such as McClellan and Burnside School. Even though the Sheffield site is consistent with the plant use in Oneota, And having maize, squash, beans, sunflowers, and chenopodium, it does not have the high numbers and weights found in the villages as of that in the Red Wing region. The reason the numbers are lower could be that the Red Wing region has much higher acreages than the Sheffield site. Marsh elder is present at the Sheffield site. This is also present at the Burnside School, McClellan, and Bryan site in the Red Wing region. There is one possible seed fragment that is documented in the La Crosse locality. Sunflower seeds have 100% ubiquity at the McClellan, Burnside School, and Sheffield site has only 46. And the La Crosse locality, only one report from the Stanford district has no evidence of sunflower seeds in Bryce Prairie phase features, but from Valley View phase features, there are 3 specimens present with ubiquity of a 20% by feature. From our initial findings and knowledge, we see some more similarities of plant use that is tied to the Red Wing region more than the La Crosse locality. It does lack high density of the present in the Red Wing region, but the Nonwood plant diversity is present. But those who live at the Sheffield site had strong subsistence practices that fall in line with the Red Wing region. That's it. Thank you.